Welcome to The Big Picture. I'm Phil Arno. The subject of today's Big Picture is genes. No, not those kinds of genes. The little pieces that make your world what it is. In fact, if you are alive, and let's hope that all of you viewers out there are breathing, genes are the engine that pretty much powers everything that makes you what you are. And in the last generation, the study of genes, or genetics as it's called, has taken off like a rocket. Only this rocket is headed for knowledge. The knowledge that might make your life a little bit longer, or healthier, or sexier, or just plain old better. Unless you're a bad guy. Because as any good student of today's crime dramas is aware, do a crime, leave your DNA, and get caught. Well, maybe in the real world it's not quite that clear cut yet, but it's getting there. Gene technology, however, isn't just for humans. Plants are being altered to make them more resistant to insects and mold, spoilage. Plants and maybe animals like mosquitoes can be altered to take away their bite. And that could save millions of lives by preventing the spread of deadly diseases. We've even heard about scientists experimenting with clones. So sometimes it's a controversial subject as well. But as with all scientific progress, the intention and hopefully the result is to benefit mankind to make the world a better place for all. And if that can be accomplished, in the end, well, that's a good thing. With me to sort out some of the multitude of genetic studies that are being explored today is Dr. Richard Gronostajski, professor of biochemistry and director of the Genetics and Genomics graduate program at the University of Buffalo. And hopefully I got your name right, or close enough.
You did, Phil. Thank you very much, and I'm very happy to be here.
Well, it's an interesting subject, and I'm sure it's a subject that a lot of people out there are curious about, have kind of a shallow or surface knowledge on, because we've all seen CSI, we've all seen, the news about clones and various other gene therapies that offer us hope, for various cures to diseases. But we really don't know much about the details of what's going on in the world of genetic technology and research. And so, UB has a tremendous program, and your ranking nationally with the program is quite well respected.
Yeah. Yes, the program that I'm the director of, the Genetics, Genomics, and Bioinformatics program, is only about 5 years old at UB. But we put it together to bring together geneticists from around the university. So that includes people from Roswell Park, people from the North Campus of the university, people from the medical school, and to give them a program that all of them could participate in and share information, share ideas about the kind of research going on. So we have a very vigorous and very rich population of geneticists at the university that are working together.
Well, it's called genetics and genomics.
Yes.
What's the difference?
So genetics is sort of the traditional name for the study of genes. It's been around for, I'd say, about 150 years, actually. And it was the study of how particular individual genes affect what we call the phenotype of an organism. So the phenotype, what a scientist talks about, just means what an organism looks like and how it behaves. Is it normal? Does it have a disease? Does it have the correct number of arms and legs? That's all the phenotype of it. And so genetics is the study of how a single gene influences the properties of an organism, whereas genomics is really a field that's only come into being in the last, I'd say, 5 to 10 years with the sequencing of the human genome. So back when genetics was first being studied, all we knew is that if you had there were certain characteristics that were passed down from generation to generation, and they could be followed in a family. Sometimes it was a disease, sometimes it was a hair color, sometimes it was whether you were tall or short, and it was known that there was some factor that was inherited that was responsible for that, but what the structure of that wasn't well understood. In the 1950s, the structure of DNA was finally, which is the abbreviation for a very long word, deoxyribonucleic acid, but we all call it DNA because it's much simpler to say, and that's the information that's passed down from generation to generation. And so once the structure of DNA was known, the decision was made to try to understand what we call the blueprint of life. And that's what the DNA is that each of us carries in us. And it wasn't until 2003 when the actual full blueprint for life, or the DNA sequence, was determined for what we would call the average human being. So it wasn't for an individual, but it was for a collection of individuals. And now a lot of work is going on. Now that we know the entire, what we call the genome, we can study genomics, which is not how a single gene affects a process, but how do all of the genes of an organism, all of the mutations that each of us might contain, how does that affect our likelihood to get diseases, our likelihood to go bald as we get older, our likelihood to live a long life or a short life. So genes influence all of those properties of humans.
Now correct me if I'm wrong, but the advancement of these studies was directly tied to the development of better and stronger microscopes that you would be able to study more closely the structures?
Right. What it came down to was the development of new technologies to sequence the DNA, to actually determine, because DNA is nothing but a long stretch of what we call letters, A, T, G, and C, that are all in a long, as the diagram shows, that are all in a long double helix. And then that information is, each one of us contain about, each cell in our bodies contains about 3 billion rungs of this ladder, this long ladder of DNA. And in human beings, that's packaged up into what we call chromosomes. So you start with the DNA, that's the simplest structure, and that gets packaged up into chromosomes, and then the chromosomes are essentially read out in each cell of our body to make that cell a brain cell, or to make it a muscle cell, or to make it a blood cell. And so all of our cells have the same DNA, but different parts of the DNA, it's sort of like a library. Every one of our cells has the same library, but different books are read out in each of the cells that make that cell what it is in part of your body.
It's almost like, you know, you have 26 letters to the alphabet, but they're sequenced in different order, and that's what
they're put together in a different order, and
that spells out different words.
And in each cell, only a part of the genome is expressed. Only a part of all of the genes that we have are actually read out in any given cell type, and that's sort of what makes each cell what it is.
And you said there's this vast number that's involved. How much progress have we made in mapping the sequence?
We've made, I mean, as an example, it cost about $4 billion to generate the first human genome. It took from 1998 to 2003. So it took 5 years, and it cost $4 billion.
And that was a map of
that was a general sequence of the DNA in a human being. Now, for $1,500, you could get, or I could get, our genome sequence. We could go to the New York Genome Center, and we could, or to a private company. We could get our genome sequenced for $1,500 now. So that's how that is the orders of magnitude from $4 billion to $1,500. That's how much the price has dropped due to the development of new technologies to sequence genomes, and that's why the idea is that in the coming years, almost everyone is going to get their genome sequenced because it's going to give us information that we can't get otherwise about the potential that we have to potentially live, whether we're likely to live longer or live shorter. But with that information comes potential dangers. So, for example, it was in the early 2000s when the first laws were passed preventing discrimination on the basis of your genome. So, for example, employers cannot not hire you because you happen to know that you have a pre-existing condition for a disease, and that wasn't really thought about until we had the ability to predict that in human beings.
Well, you know, it's interesting because this is uncharted territory, and with this kind of knowledge, you know, you think of the good things, but, you know, lurking in the back is that potential for abuse or for, as they say, good intentions gone bad. So there is, I guess you have to proceed with caution.
You do have to proceed with caution. You might, you and I are too young to remember this, but in the 1920s and 1930s, there was a movement, a worldwide movement, which unfortunately many reputable geneticists participated in, called eugenics, which was the idea that we should only let the best and the brightest people breed, and we shouldn't let those of us who we didn't think were worthy to actually reproduce. And it was a dark history in genetics, actually, the field of genetics, because that idea, sometimes it's referred to as social Darwinism, sort of came out, and it wasn't really until the rise of fascism and World War II when we saw some of the evil that can come from from thinking that way, that after World War II, that whole area of that whole philosophy essentially died away for most people. There are still some who espouse those kinds of ideas, but the vast majority of us, and certainly all of the geneticists that I know, no longer think about things in those ways.
Well, you know, there's some things that were intended to help people. I mean, people back in the time that you were talking about thought that maybe a lobotomy would cure people of certain, you know, bad tendencies, and it turned out to be a disaster. I mean, it was just, I mean...
So science really is, and medicine, are works in progress. Every year, every 5 years, even every 10 years, what we thought were the right things to do a decade ago, now it turns out that with more information and more knowledge, we have to reevaluate them. So it's a...
Well, you know what? We're just scratching the surface, and that was one question. So, but that's it for this segment. We're going to come back and maybe scratch another surface in the second segment. Fascinating subject. Stay tuned. We'll be back right after these messages.